Located in the heart of India, Gwalior is a city encased in history. The hilltop fort, signifying royalty and bravery, has weathered many an attack in the past to still keep a close watch over the beautiful city. It is to the credit of the Scindia family, which has established many educational and research institutions in the Madhya Bharat region. One such institution was the Jivaji Industrial Research Laboratory, founded by the erstwhile ruler of Gwalior, Jivaji Rao Scindia, in the year India won freedom. Following the War of 1962, Jivaji Industrial Research Laboratory was taken over by the Ministry of Defence, Government of India, and rechristened as the Defence Research and Development Establishment, DRDE. Ever since, DRDE has developed as a centre of excellence with expertise for detection, protection, and decontamination against chemical and biological warfare agents. The laboratory has been nurtured by former visionary directors such as Dr. P.K. Ramachandran, Brigadier Dr. K.M. Rao, Dr. R.V. Swami, and Dr. K. Shekhar. Today, DRDE's mission is designing and developing state-of-the-art detection, protection, and decontamination technology against hazardous materials and microorganisms. In detection, identification of a chemical or a biological agent using indigenous devices in field conditions as well as in the laboratory in an accurate and sensitive manner. Protection, developing devices which can protect human population against any chemical, biological, and nuclear eventuality. Decontamination, developing portable systems which can rapidly decontaminate or neutralize chemicals and biological agents.
Welcome to Defence Research and Development Establishment, shortly called as DRDE. We work in the area of weapons of mass destruction with particular reference to defense against chemical and biological agents. We have developed a very good infrastructure in terms of the equipments and also manpower, and as on today we are one of the world-renowned famous laboratories. We have taken up several projects, massive projects, to increase the ability to have a good detection, protection and decontamination. And our mission and vision will be satisfying in the years to come, will be at par with any of the developed countries. If any problem occurs in terms of chemical and biological emergencies, we'll be there to help the nation for combating that situation.
Administratively, DRDE can be divided into three main groups, namely chemical sciences, biological sciences, and support groups. DRDE's services thus go beyond military interests. Rather, its various divisions are coordinately working for the benefit of society as a whole, including defense forces. This complex coordination is ensured by the Technical Coordination and HR Division. It interacts with technical and corporate directorates at DRDO headquarters for forwarding various projects, obtaining sanctions, and monitoring progress. Technical Coordination and HR Division. The Technical Coordination and HR Division plays a pivotal role in preparation of Research Progress Report, Annual Report, research publications, organizing seminars and symposia, preparation of forecast budget estimates, and monitoring the budgets, manpower planning and management, nomination for training of personnel, mentoring young scientists, conducting NBC courses for services, paramilitary forces and others, and keeping the director updated. Synthetic Chemistry Division. Synthetic chemistry is one of the oldest divisions and backbone of DRDE. As the name suggests, it basically synthesizes chemical molecules required in various R&D activities carried out in this establishment. It also conducts and collaborates research and development activities related to medical protection, decontamination, chemical detection, and vector control. Another important activity is conducting studies to design, develop and evaluate various chemical detection systems of defense and civilian interests. This division has under its belt the portable gas chromatograph that has been introduced into services, along with NBC recce vehicle, mobile contamination analysis stations, and the handheld FPD detector. An important achievement of the Synthetic Chemistry Division is the development of a drug against sulfur mustard, which is a weapon of mass destruction in the hands of terrorists due to its ease of preparation. DRDE worked for a long time in this program and successfully developed an antidote against sulfur mustard. The most significant achievement of this work is the identification of DRDE-07 as a promising new compound. Process Technology Development Division. The Process Technology Development Division is a separate activity that facilitates upscaling the chemical processes and collecting data for smooth transfer of technology. The thrust of the division is synthesis, handling and process development of chemicals, development of technologies and products for low-intensity conflicts, upscaling chemicals of defense interests, supramolecular chemistry, and the development of new chemical entities of medicinal importance against toxicants. The division has supported several products. These products have military and civilian applications in times of war and terrorist attacks. Protective Devices Division. At DRDE, a dedicated Protective Devices Division is formed where scientists are continuously working on developing techniques and devices for detection, physical protection, and decontamination of CW agents. They have successfully developed many innovative products to save humans, animals, and the environment from the fallouts of chemical and biological attacks and are used by the services. Prominent among them being 3-color detection paper, water poison detection kit, canisters for gas masks, biomask, escape mask, NBC temperature and pressure swing adsorption-based filtration system, portable decontamination apparatus, survival water purifier straw, and test rig for the evaluation of canister and filter. Chemical and Biosensor Divisions. The Chemical and Biosensor Divisions are two important divisions at DRDE. The Chemical Sensor Division is engaged in the development of sensors for chemical warfare agents. These sensors are useful for both defense as well as paramilitary forces. These sensors are mainly based on the state-of-the-art ion mobility spectrometry principles. DRDE is also in the process of developing indigenous sensors against nerve gas. The prototype is already developed and is being upgraded for lab application. The Biosensor Division is engaged in the development of gas sensors for chemical warfare agents and biosensors for biological warfare agents, as well as diseases like typhoid, malaria, and brucellosis. The R&D efforts of the scientists in this division has resulted in the development of air cleaning filters, which have been produced in large quantities and supplied to the Indian Navy. This division has successfully developed nerve agent detector, which can detect nerve gases at trace levels in the field conditions. Vertox Division. Vertox Laboratory was created to fulfill the obligations of an international treaty known as Chemical Weapons Convention, signed in 1993. It is the country's first laboratory to achieve the status of designated laboratory from the Organization for the Prohibition of Chemical Weapons, OPCW. This status provides the laboratory and the country to play a decisive role in proving the compliance or non-compliance to the provisions of Chemical Weapons Convention. The laboratory has to its credit several innovative analytical methods for off-site identification and markers of CW agents. Biochemistry Division. The Biochemistry Division is involved in the identification and detection of CW agents and their metabolites in biomedical samples, namely urine and blood. detection methods have been developed for the identification of nerve agent metabolites in these samples at parts per billion level, and it has been a part of the exercise carried out by OPCW for biomedical samples. Pharmacology and Toxicology Division. Pharmacology and Toxicology of DRDE is an internationally renowned division, and scientists working here shoulder the dual responsibilities of safety evaluation of toxicants and development of antidote. In-depth toxicological studies on organophosphorus compounds, blistering agents, tear gases, heavy metals, and different referred chemicals and products from DRDO laboratories, and efficacy studies of the antidotes against toxicants are carried out in this division. Extensive studies have been carried out to evaluate the pharmacology and toxicology of 2DG, which is used as an adjunct to radiation therapy, and irritants, namely CR, CN, and CS, for their use as riot control agents. Another key activity is research on cyanobacterial and fungal toxins. The extensive research carried out here has led to the development of autojet injector, which is hailed as a very important innovation. ICT-based detection system for ricin, personal decontamination kit, first aid kit, CBW, and new antidotes for arsenic and sulfur mustard. This division has excellent facilities like inhalation toxicology setup, in vitro toxicity, cardiorespiratory system, and histopathology. Experimental Therapeutics Division. The Experimental Therapeutics Division is engaged in the development of drugs against cyanide and sulfur mustard, employing in vivo and in vitro systems. Alpha-ketoglutarate, a wonder drug, has been developed as an effective oral treatment for cyanide poisoning. It is instantaneously effective, safe and stable, and has been approved by the Drug Controller General of India. Alpha-KG is considered as a prophylactic agent for firefighters, rescue operations for victims of smoke inhalation, and therapy for suicidal, homicidal, and possible use of cyanide in chemical warfare and terrorism. The Phase 1 clinical trial is completed in humans and is now ready for inclusion in first aid kit for military use. Biotechnology Division. The Biotechnology Division at DRDE basically works on two aspects: biodegradation of human waste and development of detection system for various BTW agents. Biodegradation of human waste is the major problem at high altitude, where temperature falls below zero. Almost a decade and a half ago, the division started work on developing biodigester for the degradation of human waste. The biodigester employs anaerobic bacteria and is almost maintenance-free. Large number of biodigesters installed by DRDE at high altitudes are working satisfactorily. These biodigesters are equally useful in planes, and a number of such systems have been installed in various regions of the country. The Lakshadweep Administrative Authority has also placed an order for installation of biodigesters at Lakshadweep. The Indian Railways have recently signed an MOU with DRDO to introduce biodigester in the railways. As far as development of detection system is concerned, the division has developed advanced molecular technology-based detection system for various BTW agents. Microbiology Division. Over the years, the scientists at DRDE have developed many rapid disease diagnostic and identification kits and reagents for important human and animal disease-causing organisms. The Microbiology Division has had success in developing reagents and kits for plague, typhoid, brucellosis, meningitis and diarrhea, and respiratory diseases in humans as well as animals. They have been widely used from time to time in controlling the outbreak of epidemics in different parts of the country. The scientists in the division have also developed expertise in generating monoclonal antibodies. specific monoclonal antibodies reactive to enterotoxins of E. coli and Vibrio cholerae were generated and utilized in a simple ELISA-based system for rapid identification of enterotoxin-producing organisms. A coaglutination-based typhoid diagnostic kit for identification of the causative organisms in blood samples is also a part of its achievement. Virology Department. The core competence of the Virology Department is in biodefense against viruses of biomedical importance. Under the Biodefense Program, it has developed rapid and real-time detection technologies based on immunological and molecular approaches, namely PCR, real-time PCR, and LAMP techniques. The characterizations of the viral agent is being accomplished through complete genome sequencing for trafficking the agents in case of any eventuality. Detection systems have been developed for dengue, JE, West Nile, chikungunya, and swine flu viruses. It is one of the apex laboratories of the country for viral disease diagnosis. Work is in progress for development of new generation vaccines and antivirals. Bioprocess Scale-Up Facility. Bioprocess Scale-Up Facility is engaged in large-scale production of various recombinant proteins for diagnostic and therapeutic purposes of different organisms, particularly Plasmodium falciparum and Plasmodium vivax, dengue virus, and JE virus. These recombinant proteins are being used in diagnostic kits as well as immunoprophylactic studies against various infectious diseases affecting public health as well as the defense personnel. It has a state-of-the-art fermentation as well as purification facility to produce large amount of these recombinant proteins. High Containment Facility. A high containment facility was established at DRDE in order to ensure protection of the personnel working with highly pathogenic microorganisms and also to safeguard the general environment. The high containment facility encompasses virology, bacteriology, cell culture, and genetic engineering laboratories. Entomology Division. Armed forces deployed on borders and cantonments are vulnerable to insect bites of mosquitoes, sandflies, black flies, and other blood-sucking insects such as flies, ticks, mites, and land leeches. They not only cause nuisance but also transmit diseases affecting the preparedness of the personnel. In order to prevent the transmission of mosquito-borne diseases such as malaria, dengue, chikungunya, Japanese encephalitis, and filariasis, it is essential to control these insects. After an extensive research for over a decade, the Entomology Division developed DEPA spray and cream, which is being used by the services. This division has also developed unique products like Atracticide and wool care. Electron Microscopy Division. The Electron Microscopy Division is equipped with transmission, scanning, and light microscopes with image analysis system and also with Raman spectroscope. Electron microscopy and Raman spectroscopy facilities are being used for the evaluation of ultrastructural morphology and characterization of various compounds synthesized by DRDE, other DRDO laboratories, as well as universities and research institutions from all over the country. Animal Facility. The Defense Research and Development Establishment has an animal facility with all the necessary arrangements for breeding, rearing, and maintenance of small laboratory animals. The facility is registered with Committee for the purpose of control and supervision of experiments on animals. It has an institutional Animals Ethics Committee which closely monitors the use of animals for experimental purposes. Support Divisions. The various support divisions and groups ensure that DRDE functions as a coordinated giant machine and works ceaselessly for the benefit of the armed forces as well as the civilians. These divisions need a special mention as they offer extensive support to the scientific community at the establishment. Thus, like the heart, DRDE ensures that the benefits of scientific and technological research and development in the field of modern warfare reaches the armed forces, guarding the farthest borders of our country, as well as the civilian living in the remotest places within. Jai Hind.